best way to make it interesting for all of you is for you to participate in it. So I'd be very happy if you ask questions, if anything I say is unclear, or if you want some more details. If I think that we need to discuss it in more length, I can tell you that we can talk about it at the end. So thank you, first of all, to Anna and to everyone here for the kind introduction, for the invitation to come here. I'm very excited to be here. The last time I was in Mexico was 15 years ago, and it's a real pleasure to be back. So I'd like to tell you in relatively simple words about the work that we do in the lab, and why I think the Weizmann Institute is probably the best place to do this kind of work, the work that we do, and I'll explain why during the talk. So I want to start with a little story that some of you may have even heard about. This guy that you see here in the picture is called Phineas Gage. He lived in the 19th century in the United States, and he was a railroad worker. He used to set up new tracks with his crew. And the way they did this was they'd go to an area which needed a new track, and they'd blow up the rocks so that they could make a new path for the railway. Louder? Okay. So, and they use explosives in order to make a new path for the railroad. And unfortunately for Gage, this metal rod that you see here is the metal rod that he was using to stuff the explosives into the ground. And one of those days, the action of stuffing the explosives into the ground actually triggered an explosion, which ended up shooting this rod that he's holding straight through his cheek and out through the top of his head. It's kind of a gruesome story, I know, but the miraculous thing was that after he was blown away by the explosion, he got up. He was a little bit disoriented, but he walked himself to the hospital and he got treated. And within a couple of months, he was back on the job, and everything seemed fine. But what his family and his friends started noticing were changes in Phineas Gage's personality, which started to take place approximately after he came back to his job. He became very impulsive. He couldn't control his temper. He couldn't maintain his social relationships, and he ended up being isolated from his friends and his families, and he died in San Francisco about 15 years later from complications of lung disease. But the real story was that this was the first time in which the function of the part of the brain which was damaged by this metal rod came to light. And the part of the brain we know today, based on this computer reconstruction, his skull and the rod you can actually see in the museum in Harvard. And based on the reconstruction of the path of the rod through the brain, the scientists realized that the region of the brain that was destroyed by the metal rod is the region that we call the frontal cortex. Okay, this is the part of your brain which sits directly behind your forehead, and it's the part we know today which is responsible for all of our high-level cognitive functions. The ability to generate and understand language, the ability to retain items in short-term memory, so the fact that you can remember a phone number, like a 9-digit phone number, and then dial it, is really a result of the work of the prefrontal cortex. The regulation of emotions, regulation of social behavior, all of these highly complex functions are regulated by the prefrontal cortex, and this is the region that we study. Another reason why we believe that another reason we believe this region is so important is because in many cases of psychiatric disease, people have identified changes that take place in the prefrontal cortex, and these diseases include all these disorders that were already discussed in the movies that you saw: schizophrenia, autism, depression, addiction, ADHD. All of these different psychiatric disorders are associated with changes in the prefrontal cortex. But the problem is, and this is where we start, is that we have very little understanding of how these brain regions, these complex brain regions like the prefrontal cortex and the different structures of the brain that it talks to, how they do their job. We know that if we destroy a region, then we lose a particular function, but that doesn't necessarily mean that we understand how this region works, right? If I take away a transistor from the TV is going to stop working, but that doesn't really tell me anything about what the transistor is actually doing. So the problem really is the sheer complexity of the brain, and it's been already said that the brain is probably the most complex organ in our body. I think that's an understatement. It's really extremely complicated, and what I'm showing you here is an actual image of a very, very tiny part of a mouse brain. And what you see in these colored objects are individual neurons. So each one of these neurons is like a tiny computer. It gets its input, it listens to about 10,000 different neurons, and it somehow integrates all of those information and generates an output, which goes to an equal number of neighboring neurons. And how each one of these neurons works is already very, very complicated. If you think about each one of those neurons making 10,000 connections and trying to listen to all this information and make sense out of it, that's a very complicated task. Yeah, so we're really, like someone said in the movie, we're in the dark ages in neuroscience. We've just been able to develop the tools that allow us to ask this kind of question. And I think it's an excellent question, and we have a project which is exactly about this question. We have a project that uses the same kind of cognitive paradigms that I showed you in order to understand the regulation of attention in the mouse brain. I think when we understand something about that, we can use mouse models of ADD that have genetic mutations that lead to ADD and try to understand how these mechanisms actually change in disease, but we're really just in the beginning of that. Between the hippocampus and the frontal cortex, we can actually eliminate this process of the memories going to the frontal cortex, but we don't have a good basic understanding of that. So we're collaborating with the lab at the Weizmann in our department, who's doing functional imaging of the frontal cortex and the hippocampus, and we're trying to look at both structures together to understand how this process actually happens. And as you probably know, the hippocampus is the first region which is targeted by Alzheimer's disease, by the deposition of amyloid plaques. And we believe that if we understand the basic mechanisms of how memories are formed and how they're transmitted, we might be able to understand Alzheimer's disease a bit better.
Okay, thank you.
To measure neurotransmitter. Yes.
Yeah.
Yes, because I know that many of the illness or what you were talking about, it's maybe a lack or more than enough of neurotransmitter. Can you measure?
And they range with different specificities in time and in space. So you can measure, you can use magnetic resonance imaging to record the amount of a particular neurotransmitter, but that has a very coarse resolution. You can insert a probe into the brain which samples the brain. It's called microdialysis, and that has a resolution of...
How does it go?
Microdialysis.
Okay.
And that has a resolution of minutes at best, but we know that the brain operates on milliseconds and not minutes. So what we're doing in order to understand the release of neurotransmitters is we have an optical technique which allows us, using the same kind of implanted fibers, instead of activating the cells, we actually record their activity through the same fibers that we use for optogenetics. And I think that's a really interesting technique that we're developing. So I told you about the amygdala and the prefrontal cortex. What we can do now is we can measure the release of neurotransmitters from the amygdala to the prefrontal cortex and try to understand under which circumstances this release takes place.
It seems that when you use the transgenic mice model, it's more specific to specific neuron you want to target, and you use a specific promoter. Right. And when you use an adenovirus, it's to all the neurons that are there, right? So how do you choose which promoter you use and how you go about your research?
So in 2009, when I was at Stanford, we were really trying to get exactly at that question. And what we converged on is a method that uses the conjunction between the expression from a transgenic mouse and the injection of a virus that's only able to express in particular cells. So you might know recombinase. So we use transgenic mice that express recombinase under different promoters, and we inject viruses that can only express in the presence of recombinase. So the conjunction between the specific expression of the recombinase and the gene which is encoded by the virus basically gives us the specificity.
Great question. What is the relationship between all these studies and the regular illnesses that we have? I have a cold. Was this originated by the brain? Is it independent by the brain? I have a stomach ache. Is that created by the brain? Is that independent from the brain? How does this work?
A field which is called neuroimmunology, and I think you've probably heard about it. The brain interacts with the immune system in many different and very tight ways. So we know that the immune system can affect the brain. For example, inflammation can reduce the rate with which new neurons are generated, and that has immediate effects on the formation of memories. And on the other side, we know that stress, which is a psychological phenomenon, can affect our bodily functions very strongly. So the two systems are very tightly interacting. This is not the field that I'm studying, but I think that there's a lot to be discovered in that field.